Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Leisha Kopp from Mirus Bio.
Hi, everyone. Thanks so much for attending this presentation on achieving higher protein titers faster with the CHOgro high-yield expression system. As Leah mentioned, my name is Leisha Kopp, and I'm an application scientist at Mirus Bio. I really appreciate the opportunity to share more about Mirus and our products with you, so thanks again for tuning in. I'd like to start off the presentation by introducing Mirus Bio, our history, product offerings, and our global presence. We'll cover considerations for CHO cell use earlier in the biotherapeutic protein production process and how the CHOgro high-yield expression system makes that possible. We will look at the process Mirus followed to develop the CHOgro high-yield expression system, as well as the benefits of choosing CHOgro for your protein production platform. Following the presentation, we will have a Q&A session, so please feel free to type questions in the chat during the presentation, and we will get to as many as possible after the talk. Mirus Bio is located in Madison, Wisconsin, and was formed in the mid-1990s by 3 researchers at the University of Wisconsin-Madison with a passion for gene delivery both in vitro and in vivo. Our transit LT1 transfection reagent was introduced in 1996 and was the first serum-compatible DNA transfection reagent on the market. Mirus also commercialized reagents for small and large RNA delivery, as well as solutions for electroporation with our Ingenio electroporation solution and EasyParator instrument. Our Transit X2 dynamic delivery system is perhaps the most broad-spectrum chemical reagent we offer, as it performs well with a wide variety of cell types for applications like gene expression, RNAi, and CRISPR-Cas gene editing. The Transit virus gen reagent was developed for high-titer lentivirus and adeno-associated virus production, and along with our research grade and select versions of VirusGen, we will launch VirusGen GMP reagents and GMP grade LV and AAV enhancers in 2021. And last but not least, Transit Pro transfection reagent and the CHOgro high-yield expression system were designed specifically for protein production, and these are the products that we will cover today. I'd also like to highlight that Mirus has over 30 trusted distribution partners supporting over 40 countries around the world in all major markets. We also offer international direct ordering to countries for which we do not currently have a distributor. So if you're interested, you can find our list of international distributors at mirusbio.com/distributors, or you can always contact our technical support team with questions. In this first section, we'll discuss the drug discovery and development process for protein biologics and where the 2 primary cell types used for protein production, so HEC293 and Chinese hamster ovary or CHO cells, are used in that pipeline. We will also cover why switching to CHO cells earlier in the process offers advantages to medical researchers. I do want to take a moment to highlight the growing use of biologics for therapeutics, which inevitably impacts the drug development process as there's a continuous pressure to develop more effective molecules in the most streamlined manner possible. This figure comes from an article published in February of 2020 in Nature Biotechnology titled Fresh from the Biotech Pipeline 2019, and shows the number of new molecular entities and biologic drugs approved by the U.S. Food and Drug Administration since 1998. So NMEs are pharmacologically active compounds that are typically chemically synthesized and well-defined. Biologics, on the other hand, include products derived from living organisms such as vaccines, allergenics, cells, gene therapies, and recombinant therapeutic proteins. While the overall number of total FDA approvals has remained relatively constant, it appears that more and more biologics are being considered each year. And of the biologic drug approvals, which are designed for the treatment of diseases ranging from sickle cell anemia, macular degeneration, breast cancer, osteoporosis, and lymphoma, half are recombinant proteins, including monoclonal antibodies, antibody-drug conjugates, and other non-antibody proteins. The vast majority of protein biologics are manufactured in mammalian cells, including HEC293 and CHO cells. Here we are looking at the drug development pipeline from early discovery and target ID to commercialization. Early on in the process, and potentially through the lead optimization phase, HEC-293 cells are commonly used to produce proteins. But CHO cells become the cell factory of choice once the candidate selection process is truly underway, and certainly once the potential drug has reached preclinical and early phase clinical trials. And CHO cells are used for many reasons, including their capacity for high-density growth in chemically defined media, their human biosimilar post-translational modifications, and their strong history for regulatory approval by the FDA. So why use HEC-293 cells early in the drug discovery stages at all? Why not just start in CHO cells? Well, the answer really boils down to stealth. Prior to the introduction of more advanced CHO-based transient expression systems just a few years ago, HEC cells far outperformed CHO cells in terms of the ability to generate enough protein for testing at a much smaller scale. And HEC cells did this much faster than CHO cells. With CHO cells, in order to generate a large amount of protein, stable cell lines needed to be generated. And we're looking at this very simplified schematic of a stable cell line generation process here. The first step is transfection of the gene of interest into CHO cells, which is the short and simple part. However, you then need to add an antibiotic to the culture to select for cells that stably incorporated your construct into their genome. The rate of genome integration is very low, so most of the cells are going to die off within the next few weeks. Then comes the process of selecting for high-expressing clones and expansion of those clones to a usable culture volume. In total, this process can take 8 to 12 weeks, and to state the obvious, that's quite a bit longer than a transient production run in HEC cells, which takes 6 to 7 days. So as I mentioned before, it was only a few years ago that high-titer transient transfection systems in suspension CHO cells became available. Mirus launched the CHOgro expression system in 2015, which enabled multifold higher titers over existing CHO systems with a workflow very similar to HEC cells. This is an image of the CHOgro system production process, where usable amounts of protein are generated within 7 to 14 days. Because this protein is generated in CHO cells and more closely resembles the final protein drug product, which will be manufactured using a stable cell line much further down the pipeline, it can be trusted for efficiency, efficacy, aggregation, and glycosylation studies. With improved transient CHO expression systems, milligram to gram quantities of proteins can be generated within a couple weeks to enable the use of CHO cells for the entirety of the drug development process. And today, we'd like to share a few of the products that Mirus offers for transient protein production, including our Transit Pro transfection reagent, which is designed for transient transfection of both suspension HEC and CHO cells, and of course, the CHOgro high-yield expression system, which is an improvement upon the original CHO system I mentioned before, as it offers even higher transient protein yields in a shorter amount of time. In this next section, we'll discuss the process that Mirus scientists followed to develop our next generation transient CHO system, the CHOgro high-yield expression system. We really had 3 central goals in mind for the development of the higher yield system. First, and perhaps most obviously, we wanted a final system that offered significantly higher titers than the original system. We also wanted to keep the workflow simple with as little culture manipulations post-transfection as possible. And we also wanted to find that any enhancers or process changes added into the system would not negatively impact cell culture viability. Because the Transit Pro reagents and the CHOgro expression media provided an excellent base for this development project, our primary focus was on screening compounds to improve expression of target proteins post-transfection. We started this by delving into the literature as well as testing libraries of internal compounds to see what moved the needle, so to speak, for production levels. Of course, we then tested for titer improvements in our existing system, as well as benchmarked against competing systems. The next part of the process involved optimization of critical cell culture parameters, including cell density, complex formation conditions, timing and volume of the enhancer addition, and shifting cultures to mild hypothermic conditions post-transfection. Once we had our protocol mostly established, we tested for robustness by evaluating various types of molecules and additional cell types beyond CHOs. As I mentioned a few slides back, one of our primary goals was to improve titers over the original CHOgro system. So here we show the impact of our final enhancer compound, aptly named the CHOgro titer enhancer, which offers a twofold boost in titers over reagents alone conditions. When comparing against 25 kilodalton linear PI, where we saw a threefold difference versus the original system, we now see a six-fold higher titers when utilizing the titer enhancer post-transfection. We all know that with cell culture, the modification of one parameter will impact how every other part of the system functions. This is why product development is often a cyclical process that requires testing of every critical component and parameter individually and in concert with other critical parameters to see how they impact the system as a whole. Here we look at the impact of cell density, enhanced addition, and temperature shift on an overall production, on overall protein production with the CHOgro high-yield system. In this first data set, a human IgG1 construct is delivered to cells at either 2 million or 4 million cells per mil with or without the titer enhancer added post-transfection. Cultures are maintained at 37 degrees for the duration of the culture, and though increasing cell density and adding enhancer does increase titers, it's pretty clear that we don't realize the full potential of the system here. When shifting the cultures to 32 degrees post-transfection, the titer enhancer has a greater impact, and titers are about 1.5 times higher than the best conditions at 37 degrees. Highest titers are achieved, however, when transfecting cells at 4 million cells per mil, adding the titer enhancer, and shifting cultures to 32 degrees post-transfection. So as you can see, there's a cumulative effect of these three changes from the original system that would be missed if only looking at each of these parameters individually. Another critical parameter to optimize is complex formation time, and this is perhaps the one that people forget about the most. Complex formation is a dynamic process that involves a cationic reagent and a negatively charged DNA, and so transfection particle size doesn't just remain constant over time. In fact, particles grow rather rapidly. Here we looked at the impact of complex formation time on IgG yields using CHO-S and ex vivo cells grown in CHO media. Cultures depicted by the navy and light blue lines were transfected using PEI max, whereas the black and red lines represent cultures transfected with the Transit Pro transfection reagent and enhanced with the CHOgro titer enhancer. Significantly higher titers are achieved with the Transit Pro and the titer enhancer. However, you could miss those higher titers if extending the complex formation time out too long. Therefore, we recommend a short 3 to 5 minute complex formation step with the CHOgro high-yield expression system. Another product development goal was to maintain high cell viability while reaching higher protein production levels. Here we look at both cell density, depicted by the solid lines, and cell viability, depicted by the dashed lines, post-transfection with a human IgG construct. Gray lines represent cultures transfected with Transit Pro that did not receive the titer enhancer. Red lines represent cultures where the enhancer was added. As you can see, both viability and density are essentially unaffected by the addition of the titer enhancer, and culture viability remains above or about at 80% until day 7 post-transfection. This next data set was generated for researchers that want to extend their cultures out as long as possible and require a cell viability above 70 to 80% at the time of harvest. Mirus tested many different commercial feeds with our CHOgro high-yield system to address this need and found that the Excel Advanced CHO Feed 1 with glucose from Sigma was by far the best option available. Though it's not formally a part of our kit, we do include this as an optional step in the protocol. And in short, to kind of summarize that process, we recommend adding 15% volume of the Excel Advanced feed to cultures at 24 hours post-transfection. The graph on the left shows IgG titers and cell viability without addition of the feed, and the graph on the right shows titers and viability with addition of the feed. If harvesting cultures at the 70 to 80% viability cutoff, which is at day 8 post-transfection for the no feed condition, the titer is about 700 mg per liter. When using the Sigma feed, viability remains high at day 11, and the titer at time of harvest is nearly 900 mg per liter. So now that we've talked through the development process of the CHOgro high-yield expression system, let's look a little closer at system benefits. The CHOgro high-yield expression system includes everything needed to transfect 1 liter of suspension CHO cells along with an additional liter of media for cell adaptation and maintenance. Components of the CHOgro high-yield kit include 2 liters of CHOgro expression medium and the required L-glutamine and Poloximer supplements, 100 mL of CHOgro complex formation solution, 1 milliliter of the Transit Pro transfection reagent, and 20 mL of the CHOgro titer enhancer. So please note that all components can be purchased separately except the titer enhancer, which is only sold with the full CHOgro high yield expression kit or paired with the Transit Pro transfection reagent as a transfection and titer enhancer kit. Another benefit is the simplicity of the CHOgro high yield expression system. We recommend that cells be maintained the day before transfection to ensure that they're in log phase growth at the time of transfection, but on day 0, cells should be diluted to 4 million cells per mil, and then immediately after that, cells are transfected, the titer enhancer is added, and cultures are placed at 32 degrees. With the exception of the alternate protocol using the Sigma feed, there's no need to go back into your culture until harvest. Following transfection, researchers can expect higher yields faster with the CHOgro high yield system because antibody expression happens faster than with the XPCHO system from Thermo Fisher, which allows you to harvest and analyze sooner and ultimately screen more candidates. In this figure, we compared expression of 6 unique human IgG antibody constructs with the CHOgro high yield expression system versus the XPCHO expression system. For every antibody, higher titers are obtained with CHOgro at day 7 post-transfection. This is particularly appealing for groups wanting to switch from expression in HEK cells to CHO cells because CHO enables that shortened timeline. CHOgro also offers flexibility to use many different suspension CHO cells for expression. Here we compare relative expression of 3 different proteins in the CHOgro high yield system with either CHO-S or XPCHO cells, both from Thermo Fisher. As you can see, similar titers are observed with all constructs. Other cell types that customers use with the CHOgro system include chosen GS cells from Sigma, CHO GS cells from Lonza, as well as in-house suspension adapted CHO K1 cells. Scalability is another major concern of any research team. And in this experiment, we transfected CHO-S cells growing in 2 ml cultures in non-tissue culture treated 6-well plates, as well as 20 ml, 500 ml, and 2-liter cultures in various sizes of Thompson shake flasks. We obtained very similar antibody titers across the cultures, demonstrating scalability over a 1,000-fold range. Another note to make, all transfected cultures were maintained on the same shake platform at 125 RPM at 32 degrees until harvest on day 14. Cost is another major factor when choosing a transient expression platform. Not only does the CHOgro high yield expression system outperform the XPCHO system at earlier time points, it's also roughly half the price. The list price for a CHOgro high yield expression system is $713, which includes materials sufficient for 1 liter of transfection and an additional liter of media for cell maintenance. In contrast, 1 liter of XPCHO media and 1 exfectamine CHO transfection kit totals over $1,200 at US list pricing. So again, about double the cost for lower performance at day 7 and similar performance if extended out to day 14. Though PEI-based reagents do tend to be some of the cheaper reagents on the market, protein titers are 3 to 6-fold lower than what can be expected with the CHOgro high yield system, even if transfecting cells growing in the same culture media, so in this case, the CHOgro expression media. If factoring the cost of media, labor, space, and the time needed to generate an equivalent amount of protein with these less effective reagents, the CHOgro high yield system ends up being significantly more cost effective for you in the long run. So to summarize, the CHOgro high yield expression system from Mirus offers researchers a streamlined workflow for transient protein expression in suspension CHO cells. You can expect higher titers at an earlier time point than the XPCHO system and overall higher titers than any PI-based reagents. CHOgro is scalable and allows you the flexibility to use many different suspension CHO cells, including CHO-S, XPCHO, and chosen cells. It's also cost-effective and requires no commercial license for R&D or manufacturing use. So if you're interested in evaluating the CHOgro high yield expression system, you're welcome to request a sample through our website at mirusbio.com/sample. So you'll just complete the form, verify your shipping information, and submit the form to our technical support team to review. Once verified, your samples will typically be shipped out within 48 hours, so you receive things very quickly. Thanks again for attending our webinar today. If you have any questions about the CHOgro system or Mirus in general, please do not hesitate to reach out. My email address is listed on the left side of the screen. Our fantastic technical support team is also available at the email address and phone number listed here. If you prefer text-based conversations, however, you can chat directly with one of our scientists through our website as well. So this concludes the slide portion of our webinar, but please stick around for the Q&A session as well. Thanks again.
Okay, great. So thanks, Leisha. So before we get started with the Q&A, go ahead and start entering your questions into the chat question and answer. But we're going to go ahead and we have some questions for you really quick. They'll appear on your screen. So the first question is, what aspect of your current protein expression platform would you like most to improve? Please select one of the following. And then as you scroll down, the second question is, which of the following do you perform? Select one of the following. The options are transient transfection for biotherapeutic production, transient transfection for mechanistic studies, stable cell line generation, other, or I do not perform transfections for protein production. And the last question, all the way down at the bottom, if you can scroll down, is what types of molecules do you express in your lab? Select all that apply. Monoclonal antibodies, Fc fusions, bispecific antibodies, antigens, non-antibody antigen proteins, or I do not currently produce proteins in my lab. You can go ahead and answer those questions. We will go ahead and get started with the Q&A shortly. I'm just going to give you a couple more seconds. Just we can see how many of you have answered, so we're just trying to make sure that we get all those answers before we remove the question from your screen. And then we will start with the Q&A. Second. So it looks like we have a couple questions in the Q&A. Excellent. Okay. So Leisha, are you ready for the questions?
I am, yeah. Let's go for it.
Okay. So if your transfection reagent is used with my company's cell growth medium, are there any medium components that interfere with the transfection process?
So that is a great question because there are definitely medias out there that aren't compatible with transient transfection. So the transit pro reagent and the enhancer can be used with a lot of different serum-free cell culture medias. We've certainly tested them in many before we came up with the CHOgro formulation. But yeah, a lot of the medias that are designed specifically for expression in stable cell lines, for example, do have additives that tend to either knock down or completely abolish transfection. And if this is the case with your media, so definitely test it. And if you find that that is true, it's very possible to grow cells in your media and then collect them, so spin them down and resuspend in CHOgro media, say the day before transfection, perform the transfection the next day, and then you can either keep the cells growing in CHOgro or swap back into your growth media. We definitely have customers that do that to kind of circumvent that transfection stubborn media that they're working with.
Okay. And can you comment on analytical data from the CHOgro system? What does the typical end-length glycosylation profile look like for monoclonal antibodies in the system?
Another good question. So we haven't done a lot of glycosylation studies in-house, but we've worked with other customers that do that obviously frequently. And what they're finding is that the glycan profiles with transiently expressed proteins and specifically IgG antibodies are very similar with the transient system as they are to what they would normally expect with their stable systems. So very comparable.
Okay. And does your transfection system require medium exchange prior to transfection?
Typically, no. So again, there's that caveat of if you're using, well, I guess our systems, if you're using the full CHOgro systems or you're using the CHOgro expression media in the Transit Pro and the enhancer, we don't typically recommend that you do, that you collect cells and resuspend in 100% fresh media prior to transfection. We just recommend that you maintain cells the day before and then dilute the appropriate density for the time of transfection. So no complete media exchange before or after with the system.
Okay. I'm going to break this one into two. So are most of commercial feed medias compatible with CHOgro, and is there any data to support that?
Okay. So I think I'm understanding the question. I would say it's not necessarily. We've tested a lot of feeds with the CHOgro system. We've tested feeds from most major vendors, most things that are commercially available. So Thermo, Sigma, Lonza, GE, and while we see that they don't necessarily hurt expression in any way, they're not necessarily adding a benefit either. We really did see that benefit with the Sigma feed, though. And so if you're looking for a feed, that's really a good one to start with. We've also looked at addition of very small molecules and nutrients, including glucose, and nothing really moved the bar in terms of improving titers. Some did enhance viability for a couple of days, but the Sigma feed really was the one that did both, and so we did most of our development work with that once we found it.
And then why is it necessary to add Poloximer in the shake flask for CHOgro?
Got it. So a lot of the serum-free culture medias already have that built in. So it's Poloximer or Pluronic, same base formulation, and it just really helps the cells handle like the shear forces of that suspension swirling culture. So it helps cells, grow well and not die, essentially, when they're put under the pressure of a moving culture.
You can go ahead and type your questions in. As a reminder, only Leisha and myself will see your questions. So the next question is it difficult to adapt cells to the CHOgro media? What process do you recommend?
Right. So typically, no, it's actually very simple to adapt cells to the CHOgro media because the media is really supportive of cell growth. It's very nutrient rich. And so most of the cell types that we've worked with and that customers have worked with really do swap into CHOgro very nicely. In fact, we've done a lot of experiments where we did just a hard adaptation. So again, collect the cells, resuspend and transfect, resuspend in CHOgro and transfect the next day. And it works most of the time very well. There are some cases where, we did see a little bit of a loss in titer. And so just as a hard and fast rule, we do recommend that you do a serial adaptation or a sequential adaptation into the CHOgro media. And that basically just means starting with 75% of your current media, 25% CHOgro, monitoring cell viability and growth, making sure that they are, at least doubling every day, that they're 98% viable before you move farther, so 50-50 for the media breakdown, and so on and so forth to 25% current, 75% CHOgro, and then 100% CHOgro. That's the safest way to do it, and so that's what we have written in our protocol. Although again, there are those cases where folks are growing their cells in a stable media that doesn't allow transfection, and they don't want to do a complete switchover, and so they'll throw in that media, swap to CHOgro, do the transfection, and then swap back.
Okay, and what vector do you recommend using with your system?
So that's going to vary depending on what you want to express. We see people using commercial vectors all over. Unfortunately, we don't have a vector set that we would recommend, again, because it varies so much and we don't sell vectors ourselves. But there are a lot of commercial options to look into.
Okay. And can you use the system with HEK 293 cells?
So that question actually comes up quite a bit too, because it does work so well with the CHO cells. But so while the Transit Pro reagents can be used with HEK 293 cells, the suspension cells, we definitely don't recommend it. The CHOgro media or the titer enhancer for the HEK cells, the media basically doesn't seem to support 293 growth all that well, which is also going to strongly impact your expression levels. So yeah, CHOgro is definitely a suspension CHO system exclusively.
Okay, well, it looks like we just have time for one more question. If we didn't get to your question, we'll be providing all these questions to Leisha, and she can follow up with you directly after the webcast. So the last question is, what kind of plates do you use for 2 milliliter scale experiments?
Got it. So we've used very, different vendors for this. So, Corning plates and other types of plates. And any of them work fine. The thing to keep in mind is that they need to be non-tissue culture treated so that the cells don't adhere to the surface. But otherwise, most 6-well plates are built very, very similarly with about the same depth and width. So you can use any of them. Just make sure that when you're using that smaller scale, that you have a shake speed that works well with that plate, and that's outlined in our protocol too. But just make sure the cells are growing nicely and viability remains high, and you should be okay.
Okay, great. Thank you, Leisha.
Thank you.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.